Thank you very much. It's an honor to be the introducer of this little RNA section in this conference. And what I'm going to try to do as I go along is to try to make as many connections as I can with the world of proteins, because it's where the majority of the people here come from. The question that I'm interested is, when can we tell that an RNA has a structure or not? Which is a slightly different question than the one that one approaches when you enter one of these casts of RNA puzzle competition, in which you know that the sequence that you are given is a structural RNA because you know that somebody has already found the structure. You are just trying to get that. So statistically, there are two different questions. So to get started, so here you have a structural RNA, a really interesting one. It's called RNA-sp. RNA-sp is in charge of carrying the ends of another structural RNA, the tRNA. So when tRNA is transcribed, it's much longer than the actual size it has, and RNA-sp comes and trims the ends of it. And one important thing about this structural RNA is that the RNA part is the catalytic component of it. So which is something that deserves somebody a Nobel Prize when they first find that RNAs by themselves could be catalytic. So what I'm showing you here is the 3D structure of it. So you see the helices, that different helices are color-coded differently, and one of the characteristics of RNA 3D structure is that the helices tend to stack on top of each other. That's what is called coaxial stacking. And if I take this 3D structure and I kind of put it flat in a plane, what I get is what you have here on the right-hand side, which is the collection of base pairs that produce these helices. That's what in RNA we call the secondary structure. So right off the bat here, you see already two important differences between RNA and proteins. First of all is their size. So this structure is not just the RNA component, which is here. This little thing over here is the RNA-sp protein. So in bacteria, there is one of each, RNA-sp protein and RNA-sp RNA. And you see here how the size of the protein is about a third of the size of the RNA. And that is typical. And the other thing that you also can see is that the secondary structure of this RNA is non-local. The very first residue in the 5' end of the molecule is base paired into a residue that is really almost at the other end of the molecule. And this is a standard of RNA. Differently, to get the secondary structure of a protein, that is very local. You can see that within the backbone. Okay, so those are two differences of that. So another important thing of this RNA is that it's evolutionary conserved. RNA-sp appears in many different species, all the way from bacteria to eukaryotes, and that reflects on a pattern that you observe in alignments. So this is just this little, I'm just taking this little hairpin over here, and I'm going to a deep alignment of this molecule, and I've just taken 9 cases. So this is the molecule for 9 different species. And what you observe is that, so here are the base pairs, is that for each of the base pairs, you observe a pattern of variation that preserves the base pair. So I have color-coded in green the two positions when they have changed with respect to what is the majority of it. So for instance, for this one is a GC, here it has changed to UA, and here it has changed to an AU. Those are changes, the both residues have changed, but in a way they preserve the base pair. Watson-Crick base pairs are typically AU, CGs, and GUs, some other exceptions. And you also have cases like this case over here, in which you have a base pair that is somewhat unfavorable. That also happens. But overall, you see a picture of variation that preserves the structure. And that is typical of any RNA, a structural RNA that is evolutionary conserved. So how do you measure this covariation? That is usually very easy. There are many different methods. One of the typical ones would be mutual information. The one that I'm favoring for the method that I'm going to talk to you about is this one called GTEST, which has the same form of mutual information, but instead of measuring probabilities, it measures observed counts. So the only difference is that if I've had two pairs of positions that have the same probabilities, they would have the same mutual information, but if with one of the pairs have fewer gaps in the alignment than the other, it would have a higher g-test, which I think is a property that you want to preserve. And you could use any other measure of covariation that you want. You could use something based on POTS models, like the ones that people have been using, or this could just be some complicated neural network that you are feeding to the system that at the end of the day gives you, for a given position, the probability that is covariant with any other position. So whichever weight you use, that would measure the covariation in the alignment. And historically, covariation has been extremely useful, helping us to get structures. So I'm giving you here, this is the structure of the whole ribosome, of a bacterial ribosome. The blue is the small subunit, the white is the large subunit, and again, the proteins come here as little Christmas decorations that you have on top of the molecule. So there are about 50 proteins associated to this structure, and these are the two larger structural RNAs that we have. These were the crystal structures of these appeared around the year 2000, and just a couple of years before that, the structure, the base pairs were obtained by covariation analysis with an accuracy of over 98%, which is really amazing. And that is because ribosomal RNA is such a conserved molecule that you can have really beautiful alignments for it. But the question that I'm asking here is slightly different, because all the previous cases is like, you have an RNA, a structural RNA in your hands, and you wanted to see what the structure was here. Now the question is, I have a transcript, and I want to know, does this have a structure or not? And this is important because of the landscape of eukaryotic transcription, which I'm going to try to briefly describe to you over here. So eukaryotic transcription, you can basically divide it in two: transcription by the RNA polymerase 1 or 3 or polymerase 2. So all the structural RNAs that we know and love are transcribed by pol 1 or pol 3. All the RNAs that you have in the CASP or puzzle competition, unless they are synthetic, they're probably falling here. And these are all the well-known structural RNAs. And then you have transcription by polymerase 2 that includes all the protein codings are transcribed by pol 2. So in terms of genomic coverage, all these RNAs only cover about less than 1% of the genome, while all protein coding genes cover about 40%, at least 40% of the genome. But instead of RNA that you observe in cells, 90% of the steady state RNA in a cell is of this kind versus 10% that is of the protein coding kind. Now, this is at a steady state in which is a balance between synthesis and degradation. Now, with new transcriptomic methods, we can see nascent transcription before degradation has any chance to act. And then when you look at that, you observe that this 90% steady state is only 30% of the transcript. So there are about 70% of the transcript as they come out that belong in this class. So what are all these things? Okay? Because many of them are associated to protein coding genes, and they could be the result of premature transcription termination, read-through, and many other effects that they might be functional or not. So one interesting question is, like, people ask, what are all these non-coding? Usually they go by the name of non-coding RNAs, and one asks, what are they do? Are they functional or not? And I think there are going to be many different things here, anything from they are just byproduct of protein coding transcription to they are functional by an RNA structure. So I'm trying to find which possible small subset of these transcripts have a structure that I can believe, so that I can continue doing experiment of those. So that's the goal of this. And because once you have all these transcripts in hand, it's very easy to produce a structure. It's very easy to use a computational method, to use some chemical probing and say, oh, this is the structure. So these are several examples of different long non-coding RNAs. You might have heard or not their names, like hot air X6 is a really good one, cool air in plants, for which people have proposed structures and they have said, oh, these are evolutionary conserved structures, thus this is doing something important. So the problem is that it's very easy to produce a structure for an RNA. So here, traditionally we have, since the 80s, we have methods that you take a sequence, you can produce a structure. So here I'm giving you, this is a real structural RNA, a SAM riboswitch, and just I'm giving you predictions. So these are different packages that give you structures, predictions of the structures for just giving the sequence for that. So you see that the accuracy is variable, even if you introduce this shape here is experimentally determined data that gives you some information about accessibility of the nucleotide. Now, so these methods are relatively accurate, but more importantly, if I take a randomized version of this sequence and I put it through the same programs, I can get things like that. Here I have selected one of those. So if you didn't know any better, it would be impossible for anyone to decide whether that this is just a random structure versus this is a real structure. And that's one of the problems of RNA structure when you try to find new structures. All right? So that said, so the question is like, most RNAs have structures because you can find it in any possible way, and maybe if you crystallize that, they would produce that structure. But is the structure functional relevant? That's what I'm trying to ask. So I'm going to try to convince you that covariation analysis, covariation information, evolutionary information in structural alignments is the way of going about this signal. Now, but you need to ask, what is all covariation due to RNA structure? You need to distinguish, because if you give me an alignment and you calculate covariation by any of the methods that I showed you before, pretty much every pair of columns are going to have some variation unless it's absolutely conserved, which is very rare. And then I'm going to try to decide, is that covariation due to RNA or is due to something else? And if you wanna try to identify structural RNAs through covariation, you need to be able to make that distinction. So what I'm going to try to convince you of is that there are different sources of covariation other than RNA structure. One of them being just simply that the sequences are related by a common ancestor. So here, this is a toy sample. This is supposed to be 2 columns. Half of them have UA, half of them have CE. So you will look, if you only see that, you will say, well, this looks like a beautiful case of covariation for a base pair. And here I have exactly the same situation for UAs and for CEs. In this case, and I'm assuming the same phylogenetic history in both cases as well. So in this case, what just happened is that early in the tree, the 2 positions independently mutated, but in a way that it appears like an apparent base pair change, because a C changes to a U and the G changes to an A. But nothing else happened in the tree. This could be just the result of 2 completely independent mutations that happened there. Versus this situation in which there are 2 double substitutions that coordinately appear at different places in the tree, preserving the base pair. So the actual output is the same in both cases, but this is a case in which you could not distinguish these from just the sequences are related by phylogeny versus this case. So that's what this tool that I'm introducing here tries to do. And it's called RSK for RNA structural covariation above phylogenetic expectation. And what it does is, given an input alignment, calculates the phylogenetic history, and it generates a bunch of what is called null alignments. And what these null alignments do is they introduce the same number of mutations for each branch in the tree. But instead of the mutations appearing exactly at the positions where they appear in the original alignment, at each branch, those substitutions are put at different positions, are put at different positions in the alignment. So all the same mutations are in any of these null alignments has the same number of substitutions that the original alignment, but if there was any correlation in the position at which those substitutions occur in the original alignment, that has been randomized. So with that, you can calculate the covariation of all base pairs here. That gives you a null distribution of covariation that is not due to RNA structure, it's just due to phylogeny. So now you come with your real alignment, you calculate the covariation of those, and you can decide which ones are significant. Okay? So that's what the method does. So using it, you can find real structures, and you actually can find more than just base pairs. So I'm showing you here again the example of the same riboswitch, the one that we were predicting very different things for very different methods. So this is the rendering of the secondary structure based on the 3D structure. So the 3D structure of this RNA is known. So you have two helices here that are stacked, two helices here that are stacked. There is a pseudonode here, and here you have this L shape that is called a King-turn. Okay? So this is how the structure was annotated in RFAM until very recently. So RFAM is a wonderful database of structural RNAs, and for each RNA you have an alignment that annotates the structure on it. So it's good. It has all the hairpins. You see in green, you see the base pairs that significantly covariant according to the method RESCAPE. But what you see is that, and this is the analysis that you do with RESCAPE. So what you see many things here. So you see that this structure is missing some base pairs because that here appear because they covary. And once you see this tells you that these two helices covary one on top, coaxially stacked on top of each other. When two helices end without any residue in between, that means that they are going to come on top of each other. So that gives you that information over here. It also tells you that there is actually a pseudonode between here and here. Okay, it also shows this kind of base pair here that covary that looks like a loose base pair. No prediction method would put a base pair between things that are by itself, and then you realize how it makes sense. That when you realize that what's happening, this is the base pair that allows these helices to come back here on top and coaxially stack with P1. So that's that base pair over there. Okay, and you also see more things that are not base pairing. So once you have this is called an internal loop, because there is no Watson-Crick base pairing in both sides, well determined by covariation, you see that there is this characteristic GA and AG. Red means that position is very conserved. When it's very conserved, cannot covary, but that is a clear signature of a motif in RNA that is called a King-turn. And once you have that GAAG, this base pair, there is no Watson-Crick, and they do not covary, but that completely identifies this King-turn over here. So covariation can tell you a lot, tells you what are the base pairs. Usually the Watson-Crick base pairs are the ones that covary the most, but they also give you information about other characteristics of the structure. So when you now run this method on the structures of some of those link RNAs that they were proposed to have evolutionary conserved structures, you realize that that is usually not the case. So here you have the case of hot air. I'm just showing you here the alignment for one of the particular helices. When you do the covariation analysis, you see that the distribution of covariation for the actual proposed base pair just falls on top of the black ones, which are the ones of the null. So that means that this tells you that there is no covariation that you could above phylogenetic expectation for any of those structures. But covariation combined with conservation give you more information, because if you have covariation, which is the case of this hairpin over here, you also have variation. Obviously, you cannot have covariation without variation. But in the absence of covariation, which is these two cases over here, you can have two different scenarios. You can have covariation. No, you can have no covariation because the alignment is just too conserved and you cannot say anything. In that case, you cannot say anything. The alignment does not give you any information for you to assess covariation. Versus this case, in which there is no covariation, but there is plenty of variation in the alignment. So that, in fact, is a signature of against a base pair, because you have enough variation. If these were covariant, you should see it already, and you don't. So that's the other piece that we add to RESCAPE. In addition to having the covariation based on some covariation measure, you have the covariation analysis with your null alignments. You also do the substitution analysis. And based on how real base pair covary as a number of substitution, you create a an L by L map of a statistical power, base pairs that have enough variation that you should detect by covariation. So using a statistical significance and a statistical power, now you can decide whether you are in the presence of an evolutionary conserved base pair, whether you are in a situation in which you cannot tell because there is no variation and there is no covariation, or whether you're in a situation in which you have evidence against a base pair because you have variation but you don't have covariation. Okay? So when you put all those two things together for any RNA, you can make this analysis in which real structural RNAs conserved are going to fall somewhere here, where this is base pairs that covary, and this is base pair expected to covary by covariation power. So this little corner over here means I can't tell you whether this is a structural RNA, a conserved structural RNA or not, and things that fall along this line are going to be RNAs in which you start to see evidence against a conserved structure, at least for the structure that you have proposed. So when you do that for all of the RFAM families, you see how you have a nice, in general, nice fit to covariation track, variation tracks covariation. You still have a bunch of alignments down here for which you don't have enough information to know for sure. But then when you compare that to the cases for the link RNAs that I showed you before, these all tend to fall along the axis of there is really evidence against having a structure over here. Okay? So what I told you so far, that just by looking at your alignment, you can have a map of what I would call positive pairs that are pairs that are supposed to be, that are part of an RNA structure, or at least they are not covariant just simply by phylogeny, and also negative pairs, which is pairs that you would expect not to be in the alignment. So using both of those, you can now make a folding algorithm. So this is a folding algorithm based on that I call CACOFOL for covariation, cascade variation covariation constraint folding, in which the goal of CACOFOL is to make a structure that includes all covariant pairs and excludes all pairs that are not supposed to be there. Okay? So in general, you cannot do that. So, and this represents different folding algorithms. I use probabilistic stochastic context-free grammars, which the standard ones do not include pseudonodes. So the covariation pairs do not have to be nested. So what I do is you split your covariation pairs in nested subgroups. Then you do your folding for each one of those, and then at the end of it, you put all the structural elements together. So that allows you to see any kind of pseudonodes, triplets, or anything, provided that there is evolutionary information for them. Otherwise, you won't see them. So when you do that, you actually start seeing pseudonodes. So this is viral RNA called xrRNA. It has two pseudonodes, one a little one here and this one here. Because there is covariation in both of them, CACOFOL is able to restore these, to predict the structure perfectly well. This is a really interesting case. This is a really large bacterial RNA called transfer messenger RNA. What this does is when an mRNA is installed in the ribosome, it brings, what's that? Oh, no way, really. I thought I have a lot more. Okay, so you can detect as many pseudonodes as you have annotated. In this case, you need 6 layers of folding to get that. You can also find long-term interactions. This is from the crystal structure. You see those over here, this helix with those. And you can also see alternative structures that happen for instance, in riboswitches. And we have used these. I wanted to get into something else. I thought I had 25 minutes. Okay. So let me go to, just to say that we are using this method to discover novel structural RNAs. And this is a particular a study in which we did a search for those in fungal genomes, and we identified 7 new ones. This is one of them that we are very interested in because it's in a 3' end of a metabolic gene, and we want to know what it does. But and this is the pipeline for that we are now working on making a tool out of it. So, but somebody asked me this morning, do you believe on deep learning for RNA structure? So that's what I would like, if I could have 5 minutes, I would like to finish with that.
We'll have a shorter break.
All right, thank you. Sorry, I underestimated the time that I had. So these are some reasons. So obviously, so this is just for the problem of RNA structure prediction. I give you the structure, I give me the sequence, I give you the structure. So there are many different methods for this case of just getting the base pairs. So there are many different methods that have come, and this is just a selection of those. If you look at their actual characteristics, they use all possible architectures that exist. The inputs have different embeddings or representations, and the outputs also are variable. Usually you get information about whether something is base paired with something else, and usually some of them need post-processing in which you need to put the information through one of the folding algorithms or not. Some of them allow pseudonodes or not. None of them allow triplets, none of them allow no canonical base pairs, and definitely not overlapping structures. So this is one of those in which you have here convolutions. They train first a pre-training on just structures, annotated structures, then on PDB. This is another one of them using transformers. But the issue of all those is that it's very difficult to test their performance, because one of the problems in many of those methods when they tested their performance is they use the same structures in testing and training. So Marcel Cisak, who is now a postdoc in my lab, he did this work before joining the lab. He actually retrained those that he could retrain, so he could only retrain 2 of those, these 2. And then he did I'm training, so he had this collection of structural RNAs, families, and then what he did is he trained on all but one and he tested on the other one. So when you do that, this is their performance, and this is compared to the performance with one of the standard thermodynamic models that we have known forever. So in almost no case, any of these methods outperforms the standard methods. I still see that this performance is not that great, but these methods have not been able to do that. So I would not say anything about 3D structure, because we have a talk right after this about that, and I think on Thursday we have another one from sorry, I forgot your name again, and about that. But I would like to finish just stressing the differences between RNA and proteins. So first of all, as you all know, we have many fewer PDB structures for RNA than for proteins. In a good day, I would say there is about 15K. Take it or give it, some methods use a few more, maybe 2,000, maybe, but that's about the number versus 100,000 that you have for proteins. Second of all, the homology is more complicated because it's a structural homology, and that means that the alignments are usually much shallower and sometimes tricky to build. Okay, and that is also part of the fact that you have a 4-letter alphabet as opposed to 20, and also because of the conservation oftentimes is at the structural level, not at the sequence level. But I think one of the good things that our RNA has that protein does not have in the same way is the kind of really deep covariation signal coming from the base pairing. I mean, there is long-range covariation information in proteins, obviously, but it's not of the nature that there is in RNA. And I hope that is a good thing that can help us with that. The other thing is that the geometry is way more complicated. An RNA, so this is just a base, a nucleotide between 2 phosphate, is much larger. For proteins, you only need 2 torsion angles to get from one amino acid to another. In this case, you need 7. And here you can tell the 2D structure from that. Here, even if you know all the position of all those angles, you have no idea whether this residue is base pair or not. You need to know to the rest of the chain to see where the other base is, to see whether it is flat with each other, whether there are right distance to call that a base pair. So those are the challenges. The 2D structure is not local. And I just will finish saying that, all the thing that I told you about today is just based on the information contained in this alignment. I hope that by using information contained extracted from many more alignments, using attention, using an RNA language model based on alignments, would help making this picture a little bit richer. And sorry about the extra time. Thank you very much. And this is my acknowledgement.
Thank you so much, Elena, for this wonderful introduction. I think it's something that kind of let us dive into, for some, a new area. So,
thank you very much. Brief question: does it make sense to look into structure of coding RNAs?
Oh, structure of coding RNAs. So that's a very interesting question. Maybe. So one thing that I didn't say is that coding RNAs have a covariation signature that is just due to coding, which is the fact that the codons, because of the codons with 6 boxes, that you have variation between first, second, and third position, and then evolution. So if you have an alignment of protein coding sequence of exons that is deep enough, we are going to start seeing covariation about phylogeny that is in between positions that are 1 nucleotide apart or 2 nucleotide apart. So that's one thing. Now, your question was about, is there RNA structure in coding regions?
There are 3D structural motifs.
They are, obviously they are. I mean, overall, I don't think, as any RNA, it can fold. If the RNA is allowed to fall long enough, probably would fall into something. Whether those structures are biologically relevant or not is a different question. And as you say, they are definitely, at least in UTRs, we know of structures. In introns, we know of conserved structures. So I don't think it's going to be all exons are going to have a structure, but definitely we should be able to find some gems hiding in there. Yes.
So, slightly related question. So, one problem you said with RNA is to make alignments. Mm-hmm. So can you extend a bit on that? What is the best tools, and how good are we, and how much we're missing?
Right.
And a related question is, GC content can vary a lot between different organisms. So I'm sure this affects alignments, like if you have GC rich or GC poor genomes. So is that also a problem? Also covariation, of course. But
yeah.
Does that matter?
So the question is about RNA alignments, and that is a really interesting topic. So the way we do, if you know that you have an RNA structure, okay, then you can make a profile context-free grammar, much as a hammer model, and that you can use to find new homologs and to align them. So if I have which is that how the Hennenfent has produced the alignments. So if you know that you have an RNA that is a structure, I would go with using Infernal to make a structural alignment, which is going to put things in the best places and you're going to see nice covariation. Now, if you are trying to align a sequence that you don't know, so you are doing an exploration, you have a UTR or a coding region, you are going to align, you are going to use that for alignment, so you need to be very careful. So I see some of the deep learning methods are using methods for RNA, or some of the 3D methods that I didn't talk about are using a method for aligning that, so that what they do is that you have the sequence, you don't know whether it's structural or not, you propose a structure. Now, using the proposed structure, you can make one of these profile models and align. That is dangerous at best, because if the sequences have low complexity and sometimes high GC is going to give you a sequence that have very low entropy, so it's going to allow you to do more of that. If you have especially sequences of low complexity, of low entropy, it's going to be very easy for these methods because you are forcing them, you are telling them, align them according to that structure. If there is enough variability, you are going to be able to do that. So that is very dangerous. So for instance, in the method that we have for trying to find novel structural RNAs, the pipeline that I briefly saw that I didn't explain, we never align using Infernal, we align using Hammer, because at least that is agnostic to any structure. So if the signal is there, you hope to capture there, but you can't force it, because if you force it, you are going to fool yourself. I mean, I have beautiful artifacts aligning, especially because if you use infernal and you use an e-value really high, like some of the methods for 3D deep learning RNA are using, then you are going to bring non-homologs. And once you do that, that is going to be a kind of work. So it's a really tricky subject.
Maybe one more question.
Hi, thanks for the talk. So I worked for a while on, protein covariation. So it was nice to see how things work on the RNA side of things. So I was curious, one of the historical problems that we had with covariation in proteins is the transitive correlations. How do you deal with that with the G-test?
The what correlation?
The transitive correlations. So for example, let's say that A and B are in contact and B and C are in contact. Then you will also observe covariation between A and C, even though they are not contacting each other.
So these methods would find them all. Okay, I mean you calculate a covariation between any position with any other position, right?
Yep.
So that's what in this case would allow us to see triplets. Okay, so sometimes you see secondary covariations as well, and those are there. And if you put them through the folding, they're going to come as an addition. So for instance, one thing that I see often, well not often, but if you have a deep of alignment, I have a hairpin. All the base pairs in the hairpin covary, okay, and I also start seeing what I call side covariation between the residues. Between one residue in this base pair and the residue across in the other base pair on the same side. I take those as secondary covariation, as you are saying, okay? But if they don't, for instance, the folding, they don't hurt the folding algorithm, because usually the ones that form the hairpin come together as nested. So they go first, they define the structure, and then the other ones are just not nested, so they just keep adding layers and layers in which you say, oh, I just have this one more, I can't extend anymore, oh, I have this one more. So at the end, you get your hairpin with your covariation, and then you start seeing all those extra covariations. So they are there. You just live with them.
Okay, well, if you can live with them, sure.
What's that?
If you can live with them, sure.
I mean, they give you information. They could give you information about a triplet. They could give you information about. They also, for instance, when you have covariation due to the fact that you have codons that occur between contiguous positions, you can have covariation between any of the three positions on the codon. So then you are going to also see that kind of covariation. That is information.
Thanks.
All right, thank you.
Thank you.